Welcome to Falcon Physician Review's Neurology Review. The next module will cover motor systems, the cerebellum. The cerebellum is an amazing structure in the brain that really helps coordinate movement. Without a cerebellum, you have major problems making coordinated movements, such as reaching or even drinking a glass of water. Here's the structure of the cerebellum. You see in midsection. It is a mid-sagittal section which is now cutting through the vermis, which I'll show you later. Right now you can see that it's off the base, off the back of the pons, and very unique and looks like almost like a fern-like structure. The cerebellum is divided mediolaterally from the vermis to the lateral cerebellar hemispheres. The lateral cerebellar hemispheres contains the intermediate zone. Here's a way that is divided. The center of that cerebellum is called the vermis, or the worm, and that is responsible for axial musculature control. The paravermis region, or intermediate zone, is just lateral to that, and that is responsible for coordination of your distal musculature. The lateral cerebellar hemisphere is the rest of that cerebellar hemisphere to the lateral portion. And there are many functions potentially of that, but most of them are unknown. There is some possible cognitive component of cerebellar function, but for USMLE Step 1, we should think of it as primarily a motor structure. So as I mentioned, the vermis is responsible for controlling axial musculature and getting information from axial musculature, as well as comparing that to what the brain wants in terms of commanding axial musculature. The lateral cerebellar hemispheres, the intermediate zone, is responsible for the distal musculature. Now, as I alluded to just now, the cerebellum is thought of as a comparator organ. It is comparing information about where the limb is in space to what the brain really wanted the limb, where the brain wanted the limb to be. And so it gets a tremendous amount of unconscious proprioception from joints and muscles. And in addition, it gets a great deal of collateral input from corticospinal tract fibers, to where the brain is able to tell that contralateral hemisphere of the cerebellum what it wants from the limb. So the cerebellum, you should remember, it controls an ipsilateral side of the body. So if I have a left cerebellar lesion, I will have coordination deficits on the left side of my body. Here's a picture of what the cerebellar peduncles look like when you cut off the back of the cerebellum from the brainstem. The inferior and middle cerebellar peduncles are the primary input structures for the cerebellum, and output primarily goes through the superior cerebellar peduncles. There are exceptions to that, but that is the main take-home message, that inferior cerebellar peduncle is input, as is middle, and the output goes through the superior cerebellar peduncles. Now, what is some of the input that comes through into the cerebellum? Well, a major portion from the inferior olive goes through the inferior cerebellar peduncle. These are called climbing fibers, and they have a very unique interaction with the cells of the cerebellar cortex. The middle cerebellar peduncle carries mossy fibers, and these are fibers from other areas, such as the corticopontocerebellar fibers. These are the collateral inputs that I talked about just a bit ago. Climbing fibers go into the cerebellar cortex, and they climb all over the Purkinje cells that are sitting there in the cortex. They have an extremely powerful influence on a single Purkinje. So one cell, one climbing fiber, may talk to a single Purkinje and have a marked influence over that one Purkinje. Another climbing fiber will synapse on a different Purkinje and have a marked influence over that one Purkinje. Mossy fibers, on the other hand, have a more diffuse input over many areas of the cerebellar cortex. Mossy fibers exert their influence via granule cells. The mossy fibers enter the middle cerebellar peduncle and synapse on the granule cells. The granule cells will then send parallel fibers into the cerebellar cortex and synapse on multiple Purkinje cells. Therefore, the influence of a mossy fiber is much more diffuse, but much weaker. Weaker, but more widespread. The cerebellum has three different layers in the cortex. The molecular layer is the outer portion of the cortex, where the fibers of the granule cells synapse on the dendrites of the Purkinje cells. The stellate cells and basket cells are also found out there. So these are some support cells of the cerebellar cortex and interneurons. Purkinje cells the Purkinje layer is made up of the Purkinje cell bodies. The granule cell layer is made up of the granule cells and then also another cell that's found there, which are called the Golgi cells. Here's a picture of the cerebellum and stained to show the amount of folia and white matter layers that are there, white matter connections to the cerebellar cortex. Here's a schematic of what it looks like when what we just talked about. So there are three different layers: the molecular layer, where the parallel fibers are synapsing on the Purkinje cell dendrites, the Purkinje cell layer, which is the cell bodies of the Purkinjes, and then the granule cell layer, where the granule cells are, and there are also Golgi cells out there. Here's a closer-up view of what that cerebellar cortex looks like, where you can again see the three different layers: molecular, Purkinje, and granule. Here's a picture of a very beautiful Purkinje cell. This is the way that these cells look. They have a markedly huge number of dendrites that have many dendritic spines upon which synapses can be made. So this is a tremendous computation machine that can take a lot of input from many different fibers. An interesting fact is that all cells in the cerebellar cortex are inhibitory except for the granule cells. The output from the cerebellar cortex goes through three deep nuclei. So it doesn't exit directly out of the cerebellum. It has to first stop in one of these deep nuclei. The deep nuclei are the fastigial, the interpositus, and the dentate. The interpositus nucleus can be further divided into globose and emboliform, but it's easiest to remember it as the interpositus. Here's a picture of the three nuclei of the cerebellum. Laterally, you see the most lateral nucleus is the dentate nucleus. The next nucleus medial is the globose emboliform or interpositus nucleus, and the most medial nucleus is the fastigial nucleus. The output from each of these different regions corresponds to the mediolateral divisions of the cerebellum. So the fastigial nucleus, being the most medial nucleus, gets output from the vermis. The interpositus, being the next one laterally, gets input from the intermediate zone. And the dentate, being the most lateral, gets its input from the lateral cerebellar hemisphere. Now we have to get information out of the dentate, out of the deep cerebellar nuclei and into the brain. How does it make it there? Well, information from the dentate goes via the superior cerebellar peduncle, as I mentioned, the primary output structure of the cerebellum, and it goes to the thalamus. It happens to end up in the VL nucleus of the thalamus. So you can think of that as VL cerebell. That will help you remember that cerebellar output goes to ventrolateral or VL nucleus. Some of those fibers on their way will stop in the red nucleus. The interpositus output goes to the superior cerebellar peduncle and also to the thalamus VL, but many more of those fibers will stop in the red nucleus and make a synapse. From the fastigial input, that information goes through the superior cerebellar peduncle, which ascends in the ascending to the ventrolateral nucleus, but it will also send an output through the inferior cerebellar peduncle. That's one of the exceptions to the fact that the inferior cerebellar peduncle is primarily an input structure to the cerebellum. But this information from fastigial can go out via the inferior cerebellar peduncle and descend to allow your body to adapt to vestibular input. The cerebellum also has three lobes that are divided anterior-posteriorly. So we'll show those now in this mid-sagittal section. The anterior lobe is seen here. The posterior lobe is comprised all the way up to where the flocculonodular lobe is. This flocculonodular lobe is a very interesting section of the cerebellum, and that it is comprised of the nodulus, which we see here, and the flocculus out here, where you can see it with the pin stuck into it. This is also known as a vestibulocerebellum. It's very important in coordinating balance and making sure that your eye movements are coordinated with your vestibular inputs. So again, balance and eye movements observed by the flocculonodular lobe. Its output goes straight to the vestibular nucleus and then can send descending information to affect posture. This concludes this section of the neurology review for Falcon Physician Reviews.